Um, I'd like to thank the organizer for inviting me to this conference. It's an honor to be here. To close our morning sessions, I want to talk a little bit more about our gray matter and its role in metabolic disease and health. So the main interest in our lab is to understand how brain controls nutrient metabolism and partitioning. Here I'm referring to an acute regulation of energy homeostasis and how nutrients move through different organs in our body that, for example, that determines, for example, how we accumulate fat in our belly or how other people develop or induce hyperglycemia and develop diabetes. And the fact that this regulation can be mostly controlled by the brain, as we and other investigators have shown previously, suggests or further underscores the role of central nervous system in the long term, not only in the long-term regulation of body weight and adiposity, but also in a more acute, instantaneous control of nutrient partitioning. And this can be also true for metabolism of amino acids, or more specifically, even branched-chain amino acids, which is going to be the focus of my talk today. So what are branched-chain amino acids, or BCAAs? They're leucine, isoleucine, and valine, and they're essential amino acids, meaning that they're not produced in our body, therefore we need to ingest it in a form of food. And they're involved in many important physiological processes like cellular signaling, protein synthesis, and energy balance, mainly by mTOR-mediated pathways. Now, about 6 years ago, Chris Newgard group at Duke University has shown that all three BCAAs in the plasma are significantly higher in the obese compared to the lean individuals. And the group also demonstrated that BCAAs and their related metabolites are strongly associated with insulin resistance. These findings are further supported by these studies in the following year that also use metabolomic platforms, suggesting that not only obesity, but also insulin resistance and diabetes are strongly associated with increased BCAA levels in the circulation, as well as their metabolites. And the association was found in Americans, as well as in Koreans and Asian Indian men and Chinese. A compelling study that links BCAAs to type 2 diabetes was recently published by Wang and colleagues, where plasma samples were analyzed from the participants in the Framingham study 12 years before the onset of diabetes. And it was not triglycerides or fasting glucose or insulin or HbA1c levels, but it was individual BCAAs that best predicted future risk of diabetes. In other words, the study showed that BCAAs are the most sensitive and the earliest marker for the risk of diabetes. In addition, a number of studies report that either amino acids or BCAAs can directly induce insulin resistance, suggesting not only correlational, but also causative role for BCAAs for the development of insulin resistance and diabetes. What's interesting is that circulating BCAAs and their derived intermediates are uniquely responsive to gastric bypass surgery. And another group has shown similar findings where while the dietary intervention induced weight loss fails, gastric bypass effectively lowers circulating BCAA levels and their derived intermediates, in this case C3 and C5 acid carnitines. So collectively, these findings implicate a role of BCAAs in the pathogenesis of obesity and diabetes and call for better understanding of how BCAA metabolism is regulated to explain why and how it is perturbed in the diabetic or the obese individuals. And this may also reveal how interventions that effectively reverses obesity and diabetes, like gastric bypass surgery, successfully restores BCAA homeostasis. So why are the BCAAs elevated in obesity and diabetes? It could be simply because the individuals take in more BCAAs because they eat more food. But circulating levels are high even after overnight fasting and even after matching their protein intake. So this is not it. Well, could it be because of increased proteolysis and or decreased protein synthesis? But a number of studies have shown that between the diabetic and the healthy individuals, there's no change in the whole body proteolysis and protein synthesis. So this doesn't explain either. And what we believe is happening is that the elevated BCAAs in obesity and diabetes are probably due to decreased BCAA degradation, leading to their buildup in the circulation. In support of this, monozygotic obese twins have decreased gene expressions of BCAA degrading enzymes in both subcutaneous and omental adipose tissues. And both obese and diabetic rodents show reduced BCAA degrading enzymes in both liver and fat tissues. And now you may ask, so what's the mechanism for this? Well, as in the case of glucose, insulin, which is the key regulator of nutrient flux, is less able to lower circulating BCAA levels in diabetics, meaning it takes a lot more insulin to lower BCAAs to the magnitude you see in healthy individuals, indicating insulin resistance. So based on this data and the literature review, we asked three important questions. Does insulin regulate BCAA metabolism? And if so, is this mediated mainly by peripheral or central action of insulin? And thirdly, is the insulin control of BCAA metabolism impaired in obesity and diabetes? And the data I'm going to show you today were recently published in Stem Metabolism. If you don't have access to it, please contact me. I'll be happy to share the data with you. So we first wanted to examine the physiological role of insulin in regulating BCAA levels in circulation. So for this, we used pancreatic clamps, where we were able to manipulate systemic insulin levels by clamping insulin at 0 minute that lasted for 2 hours. And by using this clamp protocol, we were able to maintain euglycemia while controlling the insulin levels. And we also infused glucose tracer to measure glucose flux and to confirm hepatic insulin action. So we performed clamps in male Sprague-Dawley rats at either basal, moderately high, or markedly high insulinemia. So these are hyperinsulinemic euglycemic clamps. And as you can see, the body was not different between the groups throughout the experiment. We know that we infused insulin properly because the levels were higher at both 3 and 16 millunit clamps. And again, the blood glucose was maintained stable throughout the clamp period. And the rate of glucose disposal, or RD, was also higher in the hyperinsulinemic animals, suggesting increased glucose uptake. As predicted, glucose infusion rate was higher in the hyperinsulinemic animals, and this is just the average GI calculation. And while hepatic glucose production was not different at baseline, it was significantly reduced during the clamp period, translating into increased suppression of hepatic glucose production in these hyperinsulinemic animals. So all this data confirm systemic insulin action, which is to suppress hepatic glucose production and to increase glucose uptake. Now, when we measure plasma BCAAs in these animals, as you can see, while there was no difference at baseline, there was an insulin dose-dependent reduction of plasma BCAAs in these animals, suggesting that insulin indeed regulates circulating BCAAs. Because we hypothesized that this is due to increased BCAA degradation, we measured protein expression of BCKDH, or branched-chain alpha-keto acid dehydrogenase, which is the rate-limiting enzyme for the BCAA degradation pathway that is mainly expressed in the liver. As you can see, the BCKDH protein was increased at 60 millunit, while the phosphorylated BCKDH was reduced at both 3 and 16 millunit groups. Now, in this case, phosphorylation inactivates the enzyme. So reduced phosphorylated BCKDH indicates more active enzyme, and this was further validated by increased BCKDH activity in these animals, in the liver of these animals. We also wanted to see if hyperglycemia, as seen in diabetic patients, can also regulate circulating BCAAs. So we raised and clamped blood glucose at 350 milligrams per deciliter to test if hyperglycemia per se can regulate BCAAs independent of changes in insulin. And it turns out that it does not. We also tested in a chronic type 1 diabetes model where we injected mice with streptozotocin to kill pancreatic beta cells and induce hyperglycemia. And as you can see, the animals lost body weight, and their glucose level was off the roof, and yet their plasma BCAA levels were not different compared to controls. So these data altogether suggest that insulin, but not glucose, directly reduces circulating BCAAs independent of glycemic changes, and that hyperinsulinemia causes an induction of hepatic BCAA protein, and that probably accelerates or improves BCAA degradation. This schematic shows that after ingestion of meal, the absorbed nutrients trigger release of insulin from pancreas that induce de novo lipogenesis from the adipose tissue, increase glucose uptake from muscle, and suppress glucose production from the liver. And we also have shown that our clamp data show that insulin also is able to regulate BCAA metabolism, but they don't allow us to differentiate between the peripheral versus central effects of insulin on BCAA metabolism. So to examine the role of brain insulin in regulating BCAA levels, in Sprague Dawley male rats, we implanted bilateral cannula into medial basal hypothalamus, or MBH, which is the key regulatory center for metabolism. And then we directly infuse insulin in the MBH for 6 hours, as shown in this clamp schematic. By using this clamp protocol, we were able to maintain euglycemia and also basal insulin levels throughout the clamp period. And you can appreciate that there was no difference in body weight. Blood glucose was maintained stable. And as we and others have shown previously, MBH insulin infusion increases glucose infusion rate without altering the glucose uptake or glucose disposal, leading to increased suppression of hepatic glucose production after MBH insulin infusion. And this also confirms the hypothalamic insulin action on glucose metabolism, as shown by us and many others. Now, when we ran when we did targeted metabolomics to measure the circulating amino acids and their derived metabolites, we found that MBH insulin infusion reduces amino acids in general, but the reduction was statistically significant for BCAAs. And this was further validated with a separate spectrophotometric assay that measures BCAAs as a whole. Now, interestingly, MBH insulin also lowered C3, C4, and C5 acid carnitines, which are partially oxidized intermediates of BCAAs. In support of, or supporting the concept of increased BCAA degradation in liver by insulin, there was a reduced propionate-CoA and also 3-methylcrotonate-CoA, which are partially oxidized intermediates of BCAAs, but increased acetyl-CoA after MBH insulin infusion, which is the end product of BCAA degradation. So these data suggested to us that MBH insulin or brain insulin is sufficient to regulate circulating BCAAs and their derived intermediates, possibly by inducing hepatic BCAA catabolism. And this is exactly what we saw, as I will show you in the next slide. So this is a simplified BCAA schematic depicting BCAA degradation pathway. So after ingestion of meal, the BCAAs first pass through the liver and is taken up mainly by muscle, heart, and kidney, where they're transaminated to branched-chain alpha keto acids by this enzyme called BCAAT. And afterwards, the alpha keto acids are released back into circulation, taken up mainly by liver, where they're oxidized by the rate-limiting enzyme BCAAH complex. And these acid-CoAs are further oxidized by other enzymes that finally yield either succinyl-CoA or acetyl-CoA that enter the Krebs cycle for ATP production. Now, when we did shotgun proteomics of the livers from these animals, out of about 2,500 proteins, 200 of them were found to be differentially regulated. And among them, 5 of them were found to be directly involved in BCAA degradation pathway, and they were all significantly upregulated. And these actually correspond to these enzymes in red color in this degradation pathway. And this was also confirmed by our Western blot data showing increased hepatic BCAA protein expression after MBH insulin compared to controls. So these data suggested to us that brain insulin or MBH insulin is sufficient to increase or sufficient to maintain or regulate BCAA levels and their metabolites by inducing hepatic BCAA catabolism. Now, if this is true, then the lack of insulin signaling in the brain should be the opposite, and you should raise the BCAA levels. So that's why we tested here in mice with deletion of neuronal-specific insulin receptors, or the narco mice, after overnight fasting or after refeeding for 2 hours. And you can appreciate that after overnight fasting, there was no difference between the groups, but refeeding doubled plasma BCAA levels in the narco mice compared to the controls, suggesting that this meal-induced insulin is not able to suppress the rise of BCAAs in the narcomice compared to controls because of the lack of insulin signaling in the brain. We also tested in using an inducible knockout mice model where there is a deletion of insulin receptors in the whole body, including the brain, or just in the periphery. And you can see that both models exhibit or they develop a severe diabetes. This is blood glucose level. This goes up to about 500. So they develop severe diabetes once the knockout is induced. And consistent with our hypothesis, in mice that lack insulin receptors in the whole body, including the brain, have increased plasma BCAA levels, but reduced hepatic BCAA protein expression compared to the wild types. On the contrary, mice lacking insulin receptors only in the periphery, but with intact brain insulin signaling, and hence, therefore, they have enhanced brain insulin signaling because the insulin is not being able to activate or act on their peripheral insulin receptors because it's lacking. Thereby, all of the insulin is going to the brain and therefore increasing the overall insulin signaling in the brain. And that results in reduced BCAA levels in the plasma, and it increases hepatic BCAA protein expression in the peripheral knockout models compared to the wild types. So these data suggested to us that brain insulin action, brain insulin action is required actually for the control of BCAA metabolism. 3-day high-fat feeding is enough to induce insulin resistance in the brain, at least in rodent models. So we tested our hypothesis using this model with prediction that the BCAA metabolism will be perturbed after 3 days of high-fat feeding. So after high-fat feeding, we infused an insulin in the MBH, and we followed the same clamp protocol as I showed you earlier. And as you can see, compared to the child-fed animals, the 3-day high-fat feeding impairs the ability of MBH insulin to suppress BCAA levels by 50%. Again, suggesting that brain insulin or neural insulin action is important and is required for the control of BCAA metabolism. Chronic high-fat feeding is in non-human primates, such as rhesus macaque monkeys, also has been shown to induce insulin resistance in diabetes and obesity. So we want to measure hepatic BCAA protein expression and see if that is altered. And for this, we worked with our collaborator from Oregon. They gave high-fat diet for about a year and a half, and then we measured. We got a hold of the liver, and we measured the BCAA protein expression from the liver. And as you can see, they indeed reduced BCAA protein expression in the liver after chronic high-fat feeding compared to the control monkeys. And the hepatic BCAA protein expression was inversely correlated with body weight and also with adiposity. We also see similar findings in humans, and here we had either non-obese, healthy individuals, obese, or obese and diabetic. And you can clearly appreciate that compared to non-obese, the obese or obese and diabetic male individuals, they have reduced hepatic BCAA protein expression. And surprisingly, there was no difference in females, and this may suggest gender differences in the control of BCAA metabolism. And we also show that in this case, plasma valine, one of the BCAAs, is positively correlated with BMI, but it was inversely correlated, but the BMI was inversely correlated with hepatic BCAA protein expression. And lastly, we tested in C. elegans. Now this is a really interesting creature because it has insulin receptors mainly expressed in neurons, some in the gut, mostly in the neurons. Therefore, this serves as an ideal model to study neuronal signaling or to study the role of neuronal insulin signaling in the metabolic regulation. And our prediction was that the BCAA levels will be higher in worms that lack insulin receptor homolog or DAF2. And indeed, after 12-hour fasting, when we measured BCAA levels from the worms, that is what we actually saw. We saw increased BCAA levels in the worms that lack insulin receptors in these neurons. So I hope I have shown you some evidence suggesting that brain insulin signaling regulates BCAA metabolism and that this mechanism is evolutionarily conserved from worms to mammals. So in summary, what we believe is happening is that after ingestion of meal, the pancreas secretes insulin, and then insulin goes to the brain, mostly activates medial basal hypothalamus, and that increases hypothalamic insulin signaling. And through unknown mechanism, it increases BCAA catabolism in liver, leading to reduced circulating BCAAs and their derived intermediates, mostly C5 and C3 isocarnitines. Currently, we are trying to investigate, or we're trying to identify the neural pathways that mediate the brain control of BCAA metabolism. And this includes identifying specific neuronal phenotypes in the NBH that are responsive to insulin. And also, we are trying to examine the role of autonomic outflow to the liver and how this contributes to increased BCAA catabolism. And as I depicted here, we are also interested, and we also want to apply our understanding on BCAA or on brain control of BCAA metabolism in bariatric surgery. Gastric bypass is by far the most effective treatment for obesity and diabetes today, but the underlying mechanism is far from clear. Although there are several hypotheses put out, like here, as noted here, it could be because of hormonal neural signals changes. It could be mostly in the brain, but nobody really knows why so far. And we hypothesize that gastric bypass surgery improves the hypothalamic insulin sensitivity, which in turn helps restore BCAA homeostasis. And that we also further speculate that this improved BCAA metabolism is at least partially responsible for the increase or improved glucose metabolism or glycemic control that you see almost immediately after gastric bypass, even with or independent of weight loss. And lastly, I would like to thank our collaborators, my previous mentor, Hans Rudy, and also our lab, Christoph Büttner, and various postdocs and technicians, and many more volunteers, and also our collaborators and our funding source. Thank you very much.